Hello everyone, my name is Elizaveta, and this past summer I've been working on a project called Globular Protein Vesicle Structure. I've been doing this as part of the REU in Chemical Engineering at University of Florida. So I'll start off with in general what are globular protein vesicles. So some of the components include a fluorescent protein attached with a leucine zipper, in this case glutamic acid rich. You can see to your right where it says M cherry and ZE. The ZE part is the leucine zipper, that's the coiled coils, and the globular protein is the helix that you see formed as the M cherry. Vesicles also contain elastin-like polypeptides, which are more commonly referred to as ELP, with their own leucine zipper that is arginine rich. You can see to your right where the blue part is the leucine zipper and the chain is the ELP. ELP is usually clear in solution, and any fluorescent protein you can usually see its color. So protein vesicles are formed together when the two leucine zippers come together and form a very strong bond. They have very high affinity for each other and low dissociation constant along the lines of 10 to the minus 15th. And then once they form the general structure, they form into like bundles together and end up forming a surface that forms a sphere type shape later on. So in general, how are proteins made? We start off with making a pre-culture using algae media, which you see on the left, and using a bacteria colony. In my lab, we used E. coli to put into the tube and leave in the incubation at 37 degrees Celsius overnight. Then in the morning, we would usually put it into a bigger amount of media and let it multiply even further and let it shake in the incubator up until it reaches the desired optical density. So overall, what is part of the assembly process? Some of the factors that do influence the assembly include the temperature, salt concentration, and the type of fluorescent protein used, because based on the fluorescent protein, you might need more salt in order to push it past the transition temperature at which it will form vesicles. So some of the different stages of vesicle, in order to reach vesicles, they start from soluble proteins in water, and then they form coacervates and then vesicles. Coacervates are these like bundles of the proteins strands, like with the fluorescent protein and the ELP together. As temperature increases, the ELP becomes less soluble in water, so it wants to end up further away from the water and leaves M cherry or any fluorescent protein on the outside, and helps form that basic structure that can then easily form into a vesicle. So overall, what were my project goals? I wanted to achieve small vesicles around the size of 100 nanometers. I wanted to make sure that they were stable at physiological conditions, so very low salt concentrations. I wanted to study the membrane structure and analyze the changes in the membrane structure with temperature. I was to use fluorescence microscopes and TEM and cryo-EM as some of the ways to analyze my progress in these goals. I used the fluorescence microscope just to simply confirm the size usually of the vesicles and that they were present in solution. The TEM was used to get pictures of the very small vesicles that on microscope you would just see dots, and the cryo-EM was meant to further study the membrane structure. So what in the end did I accomplish? I did reach vesicles that were the size of 124 nanometers. You can see on the sample to the right that the size that I list is a 90.85 nanometers. It was from a batch of ELPX that I did not particularly make. I did make the one that ended up with 124 nanometers though. You can see that on the picture they're quite small, even with the TEM. Some of the vesicles that I made, I did end up dialyzing, and some did survive dialysis, some did not. I did perform the TEM, and I did submit a sample for cryo-EM analysis to help see the membrane structure. Overall, I would like to thank the NSF and the University of Florida for sponsoring the REU and overall my project, and I was very thankful for being part of it. Thank you.